So it is a pleasure for me to welcome a dear friend of mine, Dr. Adetola Adesida, who I've known for more than 20 years now. He's a professor and divisional director of orthopedic basic science research in the Department of Surgery at the University of Alberta. And he leads a lot of basic scientists, biologists, clinicians, and engineers who are interested in musculoskeletal and maxillofacial cartilage tissue engineering, and also regenerative medicine research. So his research is very well funded by major organizations such as the CIHR, INSEC, the Canadian Foundation of Innovation, University Hospital Foundation, and the Alberta Cancer Foundation. Dr. Adetola completed his PhD at the University, at the School of Pharmacy at the University of Manchester, and also did his postdoc training at the Wellcome Trust Center for Cell Matrix Research in the University of Manchester and Harvard Medical School. So he's a recipient of a lot of fellowships that include the Harvard Medical School and the Marie Curie Fellowship Program of the European Commission, which he spent some time at the University Hospital of Basel in Switzerland. He's also a recipient of the Collaborative Exchange Prize of the ORS. For those who know, it's the Orthopaedic Research Society. He's also a fellow of the Royal Society of Biology and a member of the College Reviewers of the CIHR. And he serves on a lot of editorial boards, just a few here, the Nature Scientific Reports. He also holds several patents and has published a lot of papers, over 70 peer-reviewed papers in cartilage, tissue engineering, and also regenerative medicine. Thank you very much for giving us the pleasure to listen to your presentation, Adetola. It's all yours. Thanks.
Thank you very much, Professor Mwale. Thanks so much for the invitation. It's an honor and a privilege to be here today. Thank you all for attending this presentation. So I'm going to talk to you about some of the things that is ongoing in my lab at the University of Alberta, as Professor Mwale had indicated. So I mostly focus on engineering functional cartilage. I'll talk to you about some of the things that we've been doing in the area of repair and disease modeling.
Dr. Adesida, hi. Yes. Sorry to interrupt you. If you can just adjust your mic, please. This is all on this end.
Okay. Can you hear me properly now?
Yes, it's better.
It's better now?
Perfect. Okay. Thanks a lot.
You're welcome. Good to see you.
Sorry for the interruption.
No worries. So this is where my lab is located, in the Lake Sheen building. I think Professor Mwale, you've been there when you came to visit us for our graduate program. So my lab is really focused on tissue engineering, cellular cartilage-based tissue engineering. You received just from the illustration here, we would have cells being taken from patients, and then you can expand them in a cell culture, and you can put that on a biomaterial of some sort with some sort of growth factors, and engineer the cartilage graft that you want, and then you can transplant that back into a patient. The basic tenets of cartilage tissue engineering is using the cells, the biomaterial, and obviously once you get some sort of graft or implant that in a patient for reconstructive surgery. So if we can go by just thinking about the biomaterials, the biology, and the mechanics, which could be also to do with some of the areas where you want to implant the tissue of interest, for example, in the knee joints where you have cartilage, the articular surface, meniscus, for example, that will experience a lot of mechanical loading. So the mechanics is very important as well. So the vision of my lab really is to focus on replacement of cartilage and repair, as well as 3D model systems that we can use to study diseases, even something like osteoarthritis, for example. So if you think about cartilage, without cartilage, essentially, you wouldn't be alive. You'd just be a lump of gel. Most of the framework of the skeleton starts off from cartilage through endochondral ossification, where you get the bones formation from cartilage. So as you can see here, there are three different types of cartilage, three different categories of cartilage: hyaline cartilage, fibrocartilage, and elastic cartilage. And you have some in between those different categories. So I'll be focusing on hyaline cartilage as well as fibrocartilage. I won't be talking about elastic cartilage at all. So osteoarthritis, as you well know, is a big problem in Canada. About 4.6 million Canadians have arthritis. And out of those 4.6 million, 3 million of the Canadians have osteoarthritis. So it's a big portion of the arthritis that we do have. There are over 100 different types of arthritis. But osteoarthritis seems to be the most popular one. In Alberta, about 500,000 Albertans have osteoarthritis. And it is projected that by 2036, there will be 7.5 million Canadians that will have osteoarthritis. Females tend to experience it a lot earlier than men, about 10 years earlier. The cause is unknown, but there are several risk factors. As you can see here, knee osteoarthritis is a progressive destruction of articular cartilage. It leads to pain, swelling, restricted mobility. The health risk due to reduced physical activity, for example, has been ranked equally with heart disease, congestive heart failure, chronic obstructive lung disease by the World Health Organization. And 30% of arthritis patients actually have knee osteoarthritis. So it's a big problem. So as you can see here, it has been said that Canadian could lose a significant portion of its shrinking workforce to osteoarthritis unless it has developed now to sustain the economic of people who have pain and loss of function in hips and knees. So this is something that was published back in 2016. And as you can see here, the estimated economic impact, literally about 9 years from now, it's going to be about $67 billion per year. So it's a big problem. So at the heart of the matter, articular cartilage is big. As you can see here, the chondrocytes, which are the cells in articular cartilage, essentially form all the functional matrix that gives cartilage its activity. So all this white glistening material that you can see here is all matrix with a few cells called chondrocytes in between producing all these functional matrix. And obviously you can't ignore the content of water. So literally we are walking on water literally every single day, not just Jesus and Peter walking on water, but also we do walk on water because our cartilage is essentially made of, has a lot of water. The other thing that you notice here is that it has no blood supply. So it has very little reparative capacity. So something external needs to be done to build cartilage. So as you can see here, the major risk factor of cartilage for osteoarthritis is traumatic injuries. Through sports, and you can have a full condition like this, for example, and it will progress if it is not dealt with. Osteoarthritis, for example. So one of the very early interventions using the cell-based therapy is autologous chondrocyte implantation. Essentially, a healthy piece of cartilage is taken from a non-weight-bearing region of cartilage, and then the cells are harvested from that piece of biopsy. Expanded in culture for several days, and once you get enough cells, you can inject it into the full condition, cover it up with a kind of membrane. Initially, when this study was done, it was with FRSB membrane, and then it's covered up and followed for several months, and hopefully you will see cartilage regeneration in that full condition. But the problem is that the cartilage is not very good. It's actually fibrocartilage, as you can see here from this semi-quantitative RT-PCR. And you see that as the cells are being expanded, chondrocytes are being expanded in culture, they actually lose their phenotype to form the functional matrix. So you can see that collagen type 2, by the time you get to the second cycle or so, you've actually lost it. Collagen 2 has gone away. So it goes through a process that we call differentiation, the loss of the phenotype of our particular cartilage. So alternative cell sources have been wanting to be used. One of them is mesenchymal stem cells, which basically has the capacity to be differentiated towards different lineages. It could be bone, it could be muscle. But for the interest of this talk, we'll just focus on the chondrogenesis. So you can basically harvest mesenchymal stem cells from a variety of sources anatomically. can expand those cells and differentiate them towards cartilage. So as you can see here from these mesenchymal stem cells, it's a really, really interesting source for cartilage repair. You notice that in this chart in the clinical trial database, this was back in 2014 that I actually looked at this about a few years ago now. You see that 23% of a lot of the trials that's ongoing is to both with regards to bone and cartilage. So 23% of that, and I believe that this has actually increased a lot more. So in our experience in our lab, one of the things that we wanted to do was to basically harvest stem cells from sheep model, and basically expand those cells under different types of conditions, put out on the biomaterial, and then implant that to the focal defect. So this was one of our very first studies where we were looking at how mesenchymal stem cells from the bone marrow can be used to repair articular cartilage. And this study was conducted by one of my PhD students that is now practicing as an orthopedic surgeon. He is here with another orthopedic surgeon, Dr. Adetola, working on the sheep model. So one of the things that we did was to harvest the bone marrow, expand that to culture, put that on the biomaterial, allowed it to prime for about 4 days. And we did that under two different types of oxygen condition. One is the traditional 21% oxygen tension that we use in normal incubators, or the low oxygen tension, purely because cartilage exists on the low oxygen environment, as well as bone marrow stem cells actually exist on the low oxygen environment. So we actually experienced, we cultured those grafts that we would develop for cartilage under two oxygen tensions. And we used the biomaterial hyaluronan-based biomaterial, which is basically an esterified hyaluronic acid. Basically, you get that into biomaterial like this that looks almost like a cotton wool, and you can see some cells onto that. And the cells obviously will interact through the CD44 hyaluronan receptor. So we cultured that for about 4 days, and then we implanted after harvesting the stem cells. We created a defect. It's purely a focal defect to the bone layer. So purely a chondral defect, about 7 millimeters wide, and then we implanted our construct. And then we followed the sheep for about 5 and 6 months. And this is the study design. So we could do a randomization with all the N refers to the non-oxycultured ones, and the H refers to the ones that are empty conditions. And then you have the empty scaffold, and then you have also the cell-free scaffold as well. So we followed that for about 6 months. When you look at the articulated surface, after 6 months, this is what it looks like. You see that some of the focal lesions have kind of healed with time. Some are better than others. Look at histology. This is cyclonal histology. You can see the spectrum, the best, the medium, the worst. And you can see here from this small cohort of animals, we actually studied, I think it was about 5 animals that we studied in this particular study. And you can see that hypoxia culture, prime MSCs, or bone marrow mesenchymal stem cells, basically do the best repair. So in conclusion for that particular study, so we can resurface full thickness focal cartilage defects with cartilage engineered from bone marrow stem cells, and it's stable for 6 months, and there's a possible positive effect of hypoxia observed. But the study, in the context of power, is not that strong, because it's a 5 sheep study. We would like to do more studies, but this obviously needs a lot of investment. So I want to move on to the next part of my talk, which is to do with using nasal, rather than going into the knee joint or going to the bone marrow itself to harvest mesenchymal stem cell. The nasal chondrocytes from the nose, obviously, septal cartilage in the nose, can also be used to repair focal defects. As you can see from this study that was published in the Lancet back in 2016 by one of my colleagues' group in Basel, Switzerland, Ivan Martin's group, where they basically showed that they can use nasal chondrocytes to generate a graft on a type 1, type 3 collagen membrane called chondrogyne to generate a graft, cartilage graft, to repair focal defects in the knee. They basically showed that after 2 years of implantation in the focal defect, there's good cartilage regeneration based on the MRI studies that you can see. But one thing that was not very good with regards to the study was that the quality of the engineered cartilage was fairly diverse. It was very, there was really not very good grafts based on, you can see the saphenous staining here, some of the type 2 collagen. And you can see that it's not fairly homogeneous as you would expect it to be. And that's to do with some of the quality of the cells that were being used, as well as the biomaterial that was being used as well. So you don't get a full homogeneous distribution of the cartilage matrix that gives the cartilage its functionality. So one of the things that we wanted to do was to improve on that technology, just by just doing a co-culture strategy where you can make stem cells as well as mesochondrocytes together to reduce that variability that you get in the quality of engineered cartilage from mesocartilage. So we were able to demonstrate that by just mixing stem cells with mesochondrocytes, and you can get an improvement in the quality of the cartilage. And this takes roughly about 3 weeks, as you can see. So this is the engineered cartilage we were able to do for cultures. If you compare that to the cartilage that comes from the cell biopsy. So you can see that over 3 weeks, this one was taking a lifetime to make. This one will take about 3 weeks to make. So one of my postdocs was able to do that before he went into medical school. This was published in 2017. Scientific reports, as you can see here. The biomaterial that we used at the time was very similar, exactly the same, more or less the same as the one that was used by University of Basel for the study that was published in the last that I showed you. So the other thing that we were trying to do as well is to really reduce that variability that you do get, the lack of homogeneous distribution of the functional matrix of particular cartilage. So one thing that we did here was to basically compare a 3D bioprinting strategy of making cartilage grafts and compare that to using the porous scaffold, the conjugate, and see what we can make using the same donor-derived mesochondrocytes. So this is the bioprinting strategy. To mix mesochondrocytes with type 1 collagen hydrogel that we were able to purchase from Advanced Biomatrix, and then we were able to 3D print the graft and then culture for over several weeks and follow the development of the functional cartilage matrix, as you can see from this saphenous staining. We compare that to the conjugate ones here. You can see that there's a good homogeneous distribution of saphenous-positive matrix, which is basically an indication of the functional cartilage scaffold. And as you can see here, you can see that the fluorescence for type 2 collagen is pretty much well distributed in the ones that are printed through the bioprinting, compared that to the ones that are with the conjugate scaffold. And you can also see that this is the conjugate based one. If you flex it, it's like hardware, it's like the nasal cartilage in pen. Also, the 3D bioprinting one, you can also flex as well. Mechanically, you can also follow, you can see that the mechanical strength of the 3D bioprinted one tends to increase over the culture period. As the culture period increases, the mechanical integrity also increases as well. But for the conjugate one, it pretty much stays the same, regardless of whether it's a short time period or so. So that tells us that most of the mechanical strength is probably coming from the scaffold itself, the conjugate, rather than from the matrix that is being deposited by the mesochondrocyte. And the other thing as well that you can see here is that you can suture these grafts, just like Eva Martin was able to demonstrate in the trial. It was published in the Lancet that you can suture them quite nicely. You can also suture the ones that is bioprint. Just like I showed you earlier, that was bioprint. So this is after 9 weeks. You can basically put the sutures through nicely. So potentially this can also be used as a strategy to use a cartilage for reconstructive surgery. So in summary, cartilage bioprinting using mesochondrocytes as a cell source instead of going to the knee joint itself to harvest cartilage, mesochondrocytes can be used to do repair of focal defects in the joint. also make these grafts quite easily. Different sizes, 3D by printing. We're not limited by the size of commercially available conjugate size that we want. I want to move on to some of the work that my lab is doing on the meniscus. Meniscus is a fibrocartilage. It's one of the most important tissues in the knee joint to enable fluid distribution across the knee joint so that you don't have all those big stresses on the articular cartilage itself, so that you don't develop focal defects in the articular cartilage surface. So the matrix that actually makes meniscus do its work within the knee joint is produced by meniscus fibrochondrocytes. They call fibrochondrocytes mainly because they do produce some type 1 collagen, and they also produce some type 2 collagen. In terms of the meniscus, it's only about 0.5% of meniscus chondrocytes that are in the meniscus itself. So relatively very low population density of cells within the meniscus, but an abundance of matrix. Again, water is very important in terms of functionality as well. It has all these matrix components that absorb water. And it has a limited muscle, so it doesn't repair very well. It also exists under a low oxygen environment. So one of the studies that one of my PhD students, Alex, was involved in was to really look into how to engineer meniscus. And one of the things that we have to bear in mind is that this is a tissue that is mechanically loaded. It's a very, very important tissue in the context of the mechanical load functionality of the knee joint. If you don't have meniscus, you have all these focal forces on the articular cartilage surface leading to all means. You can see in this model. Now the tall meniscus here is not able to do this work, and then you have this erosion of the articular cartilage surface in this big model here. So this is a real evidence that without the meniscus in the knee joint, it's actually going to have this erosion. The picture of Roger Federer was put here mainly because to demonstrate that in 2016, when he had a meniscus injury, he could not play tennis. So for the tennis enthusiasts in the audience, well, had our technology, probably could have played the meniscus, played in the Grand Slam, but he had to go away from that because of the injury that he had. So coming back to the engineering aspects and trying to really combine some of the mechanics, understanding the mechanics of the meniscus and integrating that into tissue engineering context of the meniscus. You can see that the meniscus experiences tension on the periphery, mostly type 1 collagen bundles. It is weight bearing. You see that compression happens in the middle, in the vascular portion of the meniscus. And just taking that on board, and then also the fact that it exists under a low oxygen microenvironment. So all of that, we're integrating that into our engineering strategy for the meniscus. And as you can see by this publication back in 1980 that talked about the structure of a tissue is determined by the stress it experiences. The meniscus is pretty much shaped by the stress that it experiences in the knee joint. You see that with muscle, for example, if you want to have some sort of growth, you have to mechanical load. If you see with bone, you have to mechanical load to get growth. Nothing much is known about the meniscus in that context. So we wanted to combine mechanical load with our tissue engineering strategy, which is basically to have the cells in the meniscus, expand them in culture, and biomaterial culture, but also including mechanics. So one of the things that we first demonstrated was to engineer these constructs of the mechanical loading, as you can see here. And we were able to expose it to 30 to 40% strain at 1 Hertz, 1 hour per day under low oxygen and high oxygen environment, which is the normoxic conditions of our typical incubators. And then we can follow what is happening at the gene level to see how the mechanical loading aspects actually is altering gene expression, for example, and how that impacts on the matrix that we're going to form. And one thing that you can see immediately from when we load for a short term under hypoxic conditions, one of the things that you see immediately is that all these MMPs that are to do with inflammatory factors are actually lower due to the mechanical loading. So the good things like collagen type 2 are upregulated due to the mechanical loading. This is relative to the static conditions. You see things like VEGF, SOX9, all the good things for cartilage are upregulated. Things to do with collagen folding, hydroxylase enzyme has gone up. Head has gone up, LOX has gone up, which is for cross-linking, and then all the things to do with bad type of cartilage, type 10 and P's are going up. So you can see that mechanical loading does support the cartilage formation. And we have to ask the question, how long should we have to load for mechanical loading? And some of the gene outcomes using some of the mechanosensitive genes, responsive genes like C-force, for example, to see that our mechanical strategy is actually working, it's being transduced. You see that only a few minutes is actually needed. So some time has gone up, as well as C-force, which is telling us that there's actually being sensed by the cells. So when we load for a longer period of time, what actually happens? As you can see here, see that the three donors that we were looking at, three different constructs, mechanical strength actually increases over time. As you keep on loading, you see that the mechanical strength of the engineering is actually increases. So we can see under static conditions, the medicines that is being made is not great with dynamic compression. You see that mechanically is beginning to shape the type of tissue that we're going to get, and we think that this is going to be very important to move forward in the discuss itself before we actually. This is just to show some data that with the mechanical loading of the conditions, all the good things for cartilage formation or meniscus formation. So bringing it all together, short-term mechano-hypoxia encourages meniscus formation, gene expression. A few minutes of dynamic compression is enough for us. Long-term would be mechanical, to increase the capacity of the cartilage before transformation. These are some of the other things that we're doing to engineer full sheet meniscus before we present the sheet model to see how it performs. So this piece of news came out in the New York Times sometime late last year, which basically said that cartilage responds positively to exercise for about 10 minutes. After that, you are just accumulating more stress and damage in the tissue with no further adaptive benefits. So again, just speaking to the context of mechanical loading to the tissues, you're lowering your pro-inflammatory factors, you're engineering tissue that is much more mechanical in terms of the mechanical strength is increased due to the mechanical. So if we to move that forward into what we do for daily exercise, so we really don't need to do things where cartilage gets its benefit just by stimulation. So this was published back in the Journal of Tissue Engineering, where we basically demonstrated short time very, very good. We followed that up by another publication to refer to as mechano-hypoxia condition. So I want to talk about the molecular modeling aspects, which I mentioned earlier. What my lab is actually interested in investigating modeling sex differences, for example, in osteoarthritis. So we know that in osteoarthritis, females tend to get osteoarthritis a lot earlier than men, but more importantly, incidence and the severity of osteoarthritis is high in females. These are studies that have been published in Canada as well as in the United States. The data that we have at the moment doesn't really tell us that the hormonal differences fully explains and accounts for the disproportionation of the incidence of overweight. So there has to be some other aspects that is involved. So we were inspired by this study published by the University of California, San Francisco, where they looked at 10 patients that had healthy knee joints with no history of bone pain at all. And they had an injury in the lower extremities where they could not weight bear. But the investigators took an MRI image of the knee joints before they went weight bearing, before they went non-weight bearing on the crutches, and followed them for about 6 to 8 weeks. So they took the MRI images before and then after mechanical, after staying on crutches, they were not weight bearing. And basically what they showed was the MRI images looked very much like knee osteoarthritis when they were not mechanically loaded. And in the case studies, the authors actually indicate that this would be very important for astronauts when they go into space, because they're not able to weight bear because of the lack of gravity. So, so that really was very interesting to us. And as you can see here, these astronauts are basically floating in the air, not weight bearing, because, like I said earlier, for gravity. So mechanical unloading of the movement definitely does impact the quality, the health of cartilage. Much of the study that has been done in the context of microgravity, space microgravity, has been on bone. So we do know that astronauts do experience bone loss when they go to space, but very much is known about cartilage or even the meniscus at all. So one of the things that we wanted to do was to basically look into how the meniscus could, what the meniscus will experience if it was challenged under microgravity. We know some of the things to do with genes that are associated with healthy meniscus or healthy knee joints and osteoarthritic knee joints. And we know that the meniscus, for example, does experience some of the things that cartilage experiences in the context of osteoarthritis, like MMP going up and CON10 going up. So there's been a few studies documenting that meniscus does show osteoarthritic changes similar to cartilage. And the beautiful thing as well is that we can harvest meniscus cells from healthy patients that are gone through, maybe they have a torn meniscus, so they haven't really experienced osteoarthritis in their knee joints. So what we wanted to do was to basically engineer meniscus the way we do in the lab, and then expose that to a microgravity situation. And we can do that in the laboratory by using what we call a simulated microgravity bioreactor or a rotating wall vessel bioreactor. So as this illustration shows, when you're running your mechanical city, you're not really moving, and then obviously an astronaut in space. And this is the comparison, the parallel of what we will do in the lab, just to mimic some possibilities. So this is a study that is being conducted by one of my PhD students, really looking at how mechanical loading impact tissues in the region, this particular case with the meniscus. And basically objective for our study. So we have this meniscus cells and something else. And then we put that into our bioreactor, follow that for several weeks. This is the donor information, population levels that cells go through, the meniscus. And then we can put that into our bioreactor like this. You see that they're kind of floating in the experiencing weightlessness. So this bioreactor enables you to counter the vectors of gravity by the rotation, and then gene expression afterwards to see what is actually happening as some other analysis. And what you can see immediately here is that there are differences in the way females and the males do respond to microgravity. In particular, you will notice that there are some high responders here, like probably type 10, when helping the female population. Whereas the male, they're pretty much clustered in the same area. There are some females that basically cluster, just similarly like the male. Whereas you have these two females, for example, that basically experience a lot of hypertrophic differentiation, and we don't really know the reason for that. So we were able to stratify those differences actually using simulated microgravity. And these are some of the histology results. You can see that this is the male, the female, 6 donors, male, understudied conditions, and this is under simulated microdynamics. You can see that the type 10 collagen does vary in terms of the intensities. So one of the things that we did was to do RNA sequencing to see whether we can basically stratify some of those different populations, the high responding females and the low responding females. See that there are several genes that basically differentially express between those different groups of respondents to simulated microdynamics. And one thing that you will see from the high responding female is that things to do with ossification or things that are associated with osteoarthritis development, for example, are highly upregulated in the high responding females compared to some of the male elements of the other things that you see here, like S100. Collagen type 10 is not substantially, for example. See things to do with the gains is also upregulated, as well as ossification and weight, which is of geo profiles of the low responding and the male, they're very similar. And we were able to demonstrate that we can model some of the sex differences in osteoarthritis just using simulated microdynamics based on some of the work we've done so far with tissue engineering. So in summary, mechanical unloading of meniscus can actually help us in terms of really demonstrating biological sex differences. And we can combine that with the transcriptomics data to better understand issues of sex matters of osteoarthritis. And based on some of the written work of this publication from biocher, basically showed that stratification is really going to be very important in treating osteoarthritis patients, because not one size fits all. They're so diverse. We need to understand the mechanics. We need to understand the heterogeneity that we do see in terms of phenotypes, genotypes. So many things that we need to bring into it to understand how to treat these patients. So with that, I'd just like to wrap up and just say thanks to my funding. And I'll be happy to answer any questions.
I think you are muted, Adetola. Okay. So thank you very much, Professor Adetola Adesida, for that wonderful presentation. So I think we have a question from Raul. Please feel free to just unmute. And since I can't see some of the hands, Raul?
Yeah, a very great talk as usual, Dr. Adesida. Very informative and a very nice summary. Like, I always enjoy listening to you and stuff coming out of your lab. It's quite amazing, is the word. So I had a quick question for you. So, for example, for the role of sex and gender in the matters of musculoskeletal defects or anomalies, there is a cue for gallbladder stones. Like we say, the predisposition is 4F, say, fatty 40 female fertile. So we know that gallbladders are found more in these, if the 4Fs are kind of present. So what are your thoughts on fat, obesity, BMI, loss of estrogen, and the intra-articular fat pad, and its role in OA and meniscal degeneration?
Yeah, it's a great question. And I think we're really beginning to understand that there's so many factors that, osteoarthritis is a multifactorial disease. And there's the issues of the mechanics, there's the issue of the obesity, and there's definitely the issues of the soluble factors that are coming from a variety of tissues. And when you think about the knee joint as an organ, where you have all of these entities like the fat pad, you have the ligaments, all of them have their own things, things that they do secrete when something is going wrong. So, for example, we really don't know which tissues are affected first. in OA. So there was a publication by, I think it was a Danish group last year, that basically indicated that osteoarthritis actually impacts meniscus first before articular cartilage.
Exactly.
That was that paper. But so, and this was a tissue that was regarded as being useless for many years, just like the appendix. People are actually literally taking it out, and now orthopedic surgeons are trying as much as possible to preserve the meniscus, because now we're beginning to understand that it plays a major role in osteoarthritis, for example. And in fact, if you think about the animal model that people tend to use to study osteoarthritis, they would do what they call a DMM model, which is basically destabilizing the meniscus. So that tells you a lot that, maybe the way we need to rethink the way we are approaching osteoarthritis research. So I think the way we are beginning to really look at it, we've designed a bioreactor that actually has different chambers that will enable us to look at how soluble factors from various tissues can actually impact each other, so to speak. So we're working on that bioreactor now, and we're using our tissue engineering strategies to make all these different types of tissue within the knee joint, and then put them into the same space where they can share medium and do it in a dynamic fashion, and then we can do the readouts, whether it's at the gene level, at the protein level, whatever, to see how these various entities that you've mentioned impact, for example, cartilage or meniscus, for example, whilst we isolate them from the bioreactor. So that's the way we want to approach it. But there isn't anything that is really good out there to tell us that one thing is actually the key factor. I think we have to look at it in a combinatorial manner to be able to have a good understanding of what is going on.
I totally agree with you. I mean, I had a couple of years ago had done a study in a dog model of osteoarthritis. It was a orphan disease like mucopolysaccharidosis, where we have more gag production, but we still get OA. So in a very young animal, I was doing MRIs and trying to develop new MRI techniques. And what we saw was that we saw how fast fat pad degeneration very early on. And at the same time, we found the intermeniscus intersubstance degeneration to happen before cartilage damage.
Yeah.
So that gave us a clue that the meniscus and the fat pad are talking.
Yeah, they are.
And is the cartilage an innocent bystander?
No.
Bystander, or later on it also gets the brunt from both the tissues because they are both degenerating.
Yeah.
So it was kind of a thing that we were expecting this animal to produce more gag and be cartilage protective, but even the high gag could not protect it.
No.
And even by 6 months, we can see like specific lines coming in the how fast fat pad, and everything was just downhill from there.
Yeah.
So you raise a very good point that we have to think about it from a compartment approach.
Yeah.
And how these compartments are leaky and they, crosstalk.
Yeah, there's a crosstalk. There's an inflammatory crosstalk for sure. We do have a little bit of data on what that might look like, but I don't think we're there yet. We do have the bioreactor. We designed it. We have a patent protection on it. We're about to release it to the the rest of, so that the rest of the scientific community can make use of it, rather than just the transworld culture, which is just two tissues. So some new things will be coming out from that fairly soon. And very good point.
Perfect. Thank you.
Thank you.
Thank you. Any other questions? So one of the things you touched on was the priming of the stem cells.
Yes.
And that was quite very elegant work, and with hypoxia.
Yes.
So one of the things that we've been doing with Dr. Nicoletta Eliopoulos, who is here, an expert in stem cells, was to try to prime the stem cells.
Yes.
And one of the questions that we get asked is, how long does the priming last?
Yeah, it's a very good question. So I think I think one of the publications that one of the studies we published back in 2012 showed that if you isolate mesenchymal stem cells under low oxygen environment right from the start, when you have just a mononucleated fraction, this is before you call them stem cells, so to speak, because it's only a subfraction that accounts for stem cells. So you do your mononucleated fractionation, and then you put them under low oxygen right from the beginning. And then you can compare that to the normoxic conditions, for example. The fact that you're starting right from the beginning with low oxygen, you're probably picking those cells that are more predisposed to forming cartilage or to forming chondrocytes. And that's what we see, regardless of the donor. If you isolate those mononucleated cells, and once you isolate those mononucleated cells and develop your stem cell culture under low oxygen environment all the way through, you already primed them. And that priming, is like a selection. It's like you use the microenvironment to select them. And they're much more predisposed to chondrogenic factors than the ones that you just select, which is a mixed bag under low high oxygen environment. So that's how the priming actually occurs more. But if you were to just culture stem cells that are isolated under high oxygen, for example, typical oxygen tension of 21%, and then you prime them just for a little bit, I don't think it's going to last very long. I think it's going to revert back just based on some of the gene expression studies. You don't see the EF1 changes happening anymore. So I think it's a very short time, but if you do your selection before all the way through, then you've got a very well primed stem cells, in my opinion. Hi.
Yeah, that's great. Ovidio, you look primed.
Yeah, it's the medication, I think.
I want to ask what kind of medication.
I use a cocktail like you're using. I branch out like you do in all those things, just to increase the probability. I have a question. I have actually two questions. My first question is a question that I asked Dr. Faxon-Wale in the last 15 years. How do you differentiate yourself about when you say about tissue engineering, and then you show us a very clear thing that can be interpreted by an engineer, which is like the bioprinting, which is something clear that I can translate as an engineer. And then you delve into the very nitty-gritty of the cellular mechanism that are very hard to understand as a concept of an engineer. Yes. So where is for you the limit? So that's my first question. Yes. My second question is related to the first one, coming from kind of an engineering perspective, because I do about 3D printing. When you showed the 3D printing cartilage that you did, and I think you used a comparison with the Geistig BioGlide and with the life print. My question is and that's something that I'm really interesting. We're very tempted to kind of like perceive everything and try to extrapolate and homogenize, but every cartilage, everything, it's actually a layer of all kind of things. Yes. I will speak for the introvertible disc because I am in Dr. Wale's lab. Yes. Just the introvertible disc, when I found out how complex it is, I just realized we cannot really translate that homogeneity into characteristics that we do with homogeneous, whatever that is, printing or growing cells or whatnot. How do you see that really going long term as an application? Because short term, I think I can put any putty and it will fly. But 5 years down the road or 15 years down the road, how I will differentiate with that? And how do you see that 3D printing going in that way? Because you have a very good experience to the nanoman level to the macro level? Thank you.
These are great questions. So the very first question, the way I will address it is that I think we need to stop looking at science, engineering, and other entities of science as being separate. One of the things that tissue engineering has done for all of us is that it's brought in a variety of disciplines together, okay? Because the cells biologically are the engineers of us. They make all the things that we use. We just don't understand the cells as we should do. And I think just bringing these various disciplines together is going to help us to address very nicely what cells are actually doing. Okay, so if we just focus on just genes alone, that's not good enough. Well, what is the gene going to do? Then you talk about the protein person. That's going to say, well, proteins are the functional units of a living system. But then how does the protein actually do its job, right? And then you get into all the structure of the protein, the lock-in key mechanism and all of that stuff. So we need to understand things from different perspectives. And that's what tissue engineering is really helping us to do. So that's how I would address that. So engineering is very important. So the number of students that I showed actually today are actually not biologists. They're actually engineers. They're just people that I've taught biology. So they know how to work with cells. They know how to work with tissues, but they're actually trained as engineers. I actually like working with engineers because they're solution oriented. So that's one part. And then the second part is when you talk about the homogeneity of tissues. If you go into the body and you take a piece of tissue, there is no single tissue that is homogeneous, especially the tissues that are in a mechanical loading environment. They are shaped by the mechanics. So our job, and this is what cells do right from the beginning, when they start making all these matrices, which are actually proteins, they are shaped by the mechanics. So, for example, the blood vessels, when they start pulsating and something is flowing through them, the layers, the thin layer of cells that you see is shaped by the flow of the blood. The same thing on the articular surface. The cells that you do see on the articular surface, the matrix that they're also producing is also flat because of the articulation. But when you go into the deeper layer of the cartilage, they're kind of different. Yeah. So when we put in that homogeneous tissue that we've engineered, the rest is going to be done by the body, by the mechanical load. It's going to be shaped by that. It's going to be shaped by the different soluble factors that create a gradient to shape the cells the way they are rated. Because as you know, the diffusion of nutrients is going to be different in those layers. So different things is going to be happening. So the mechanics is going to shape it, right? So that's the way I look at it. And I think that's what we will see if we were to take an engineered graft, implant it in the patient, and then come back and do all the SEM, do the imaging tools that you want to do, analyze it before you implant it, and then take them out after implantation. You will see that it's all changed shape due to the mechanics.
Right. Yeah, thank you. I can see that you like BBC because you can explain really very eloquently. Thank you.
I was born in the US, so
Yes, yeah, no, but and it's really a very, yeah, it's a clear evolutionary concept. I can, yeah, thank you very much. Very good. Thank you.
Any other questions? So if there are no more questions, thank you very much, Professor Adesida, for that wonderful talk. And thank you everyone for coming to the seminar. And it's a pleasure to have everyone attend this seminar. So, so thank you very much, and see you soon, Professor Adesida. Thank you.
Thank you very much for having me. Bye-bye, everybody.
Bye.